Welcome back, students, to part 3. Now, part 3 is going to discuss our G-coupled receptors, and those are the majority of the receptors that are found in the body. Okay, and I want to first off talk about the structure of a G protein-coupled receptor. Remember that the G stands for globular protein. So this right here is the G protein that is coupled with the receptor. The G protein is made out of 3 subunits, alpha, beta, and gamma. The alpha subunit has a GDP attached to it. GDP is kind of like a cousin of ATP. It does store energy as well within the phosphate bonds. And you've got your transmembranous receptor. You have your G protein attached to the receptor as well. So let's talk about how the sequence of events will happen. First, your first messenger attaches to the receptor, as usual, leading to conformational changes of the structure, leading to its activation. Now, when this receptor is activated, it will phosphorylate GDP. So GDP becomes GTP, and that gives a little bit of energy to this G protein. So it detaches from the receptor. And then the alpha subunit detaches from the beta and gamma subunits. Your alpha receptor, the alpha subunit has GTP attached to it, and then it will activate another protein in the cell membrane, and that protein is either an ion channel or an enzyme. Now notice that so far, nothing has been transmitted to the inside of the cell. This whole scenario is still within the cell membrane. So it's not until the ion channel or enzyme in the cell membrane is activated and we have really not yet relayed anything to the inside of the cell. So at this point, if an ion channel is open, we are going to change the membrane potential leading to a cell response, or if we activate an enzyme, that enzyme can generate a second messenger, taking the message to the inside, leading to cellular response as well. Now let's take a look at the video to illustrate what we just talked about.
The membrane-bound receptor has a receptor site for a small molecule called a ligand exposed on the outside of the cell. The portion of the receptor on the inside of the cell can bind to the G protein, which has a guanosine diphosphate attached to its alpha subunit. When the ligand binds to the receptor site on the outside of the cell membrane, the G protein changes conformation, and guanosine triphosphate replaces the guanosine diphosphate on the alpha subunit of the G protein. The activated alpha subunit then separates from the beta and gamma subunits. This step can be repeated as long as the ligand remains bound to the receptor. When the ligand separates from the receptor site, additional G proteins are no longer activated. Inactivation of the alpha subunit occurs when its own phosphorylase activity removes a phosphate from the guanosine triphosphate, leaving guanosine diphosphate bound to the subunit. The G protein subunits then recombine and attach to the receptor in the cell membrane.
Okay, so let us move on. So this is, everything in writing. Okay, so the ligand is going to bind to the receptor, and the receptor is attached to this family of G proteins. The G protein is made out of the 3 subunits, alpha, beta, and gamma. Remember, the GDP is attached to the alpha subunit. And then when the first messenger attaches to the receptor, the receptor protein changes, and that would start the activation process. GDP is phosphorylated to GTP, and that would lead to the detachment of the whole G protein. And then the alpha subunit also detaches. Now that alpha subunit is going to activate another plasma membrane protein, and that it could either be an enzyme or an ion channel. And so it's the opening of these enzymes, the activation of these enzymes or the opening of an ion channel is what will lead to the sequence of events that happen inside of the cell. Now, since the receptor has to be coupled with a G protein, we call it a G protein-coupled receptor, and G again stands for globular, and the majority of our receptors are of these types. The G proteins, there are so many different kinds of them, okay, depending again on the ligand, depending on the cell, and what needs to happen inside of the cell. So there's several subfamilies of these G proteins. Some of them are stimulatory, and we call those GS receptors. Others are inhibitory, and we call those GI receptors. Now, how is it that we stop the action of the G protein-coupled receptor? As you've seen in the video, the ligand needs to detach, and that will break down the GTP into GDP, deactivating those the alpha globular subunit, and then everything really goes in reverse. Okay, so the whole trimeric G protein goes back and attaches to the receptor. So now that we've activated either an enzyme or an ion channel in the cell membrane, what is going to happen after that? So if that enzyme, most of the cases the enzyme is known as adenylyl cyclase enzyme. Another name for it is adenylate cyclase enzyme. So if you see either one, it's the same thing. And that is going to change ATP into cyclic or C-AMP. Well, in some cases, the enzyme is guanylyl cyclase enzyme, and that leads to cGMP, or cyclic GMP. While we said the other alternative is to open up ion channels, okay, that could lead to a variety of secondary messengers. For example, calcium ions, DAG, or IP3. If you're not familiar with DAG and IP3, that's totally fine. We'll introduce them in the next slides. So these items that we're talking about right here are these proteins, okay? So this alpha subunit we mentioned will activate an ion channel or activate an enzyme. These are the enzymes and ion channels that we're talking about. So let's see what happens if we activate either one of these two options. So let's talk about activation of adenylate cyclase enzyme, hence producing cyclic AMP, and that would be your secondary messenger in this scenario. If we activate adenylate cyclase enzyme, that would be of the GS type, so that is the G stimulatory type, and that will lead to stimulation of different things in the cell. Okay, again, your second messenger in this scenario is C-AMP. And here you can see the scenario kind of playing out. So I'll go through the whole scenario from the very top. Your ligand or first messenger combines to the receptor, changing the tertiary structure of the receptor, leading to activation of this G subunit. Since we're talking about stimulation, this is a GS protein. The GDP here will be phosphorylated into GTP, activating the G protein, and then it will detach from the receptor, and then this alpha subunit will detach from its two sister subunits, and it will activate a protein found in the cell membrane known as adenylate cyclase enzyme. Again, notice that this whole scenario is still within the cell membrane. It's not until this adenylate cyclase enzyme becomes active, it will start the process of changing ATP into cyclic AMP. The C-AMP is now your second messenger. C-AMP is going to activate other proteins, and that will lead to your cell response. So again, that little domino effect or cascade of events that happens once you've started the activation. C-AMP again is your second messenger in the scenario. So you can see here that this first messenger, one molecule of it, can lead to the production of several, lots and lots of C-AMP. All of these can activate proteins, and those can activate even more proteins, and so on. So you get the signal, what is known as signal amplification, where one molecule of your first messenger can lead to a huge response at the end. So how is it that we terminate the process? Okay, our second messenger, we really cannot do anything. The cell at least can't do anything about the first messenger because it's out of its reach. It's on the outside. What it can do, what it does have control over, is the production of cAMP. So there's an enzyme that can break down cAMP, and that's known as cAMP phosphodiesterase. It will break it down into AMP, or adenosine monophosphate. So the cell has the ability to either make cAMP through the activation of adenylate cyclase enzyme, and it also has the ability to break down cAMP using the enzyme cAMP phosphodiesterase. Know that coffee and tea that we drink those in order to stimulate our senses have caffeine in coffee, also found in tea though, and theophylline, which is found in tea. These are going to stimulate the cells through the inhibition of cAMP phosphodiesterase. Remember that the phosphodiesterase enzyme is to break down cAMP. So if you prevent the enzyme from breaking down cAMP, your levels of cAMP increase in the cell, and that is stimulatory to your cells. Now, cAMP can lead to a huge variety of different cellular responses depending on where you are in the body, using that same primary messenger. So we'll see that as an example. So epinephrine, for instance, would activate these kinds of receptors, but if it activated a receptor on an adipose cell, that will stimulate the breakdown of triglycerides. Yet on the other hand, if epinephrine attaches to a GS protein receptor in the liver cell, that could lead to glycogenolysis and gluconeogenesis. And if you're not familiar with what these terms are, glycogenolysis means the breakdown of glycogen, and gluconeogenesis means the making of new glucose. So genesis would be to create, neo means new, gluco is glucose. Okay, and so your end result of epinephrine would be to increase the glucose levels in our bloodstream in order to deal with whatever stress we are dealing with. Remember that epinephrine is one of those stress hormones. Epinephrine is another term for adrenaline, and it increases, the production of epinephrine increases due to stress. And when we're stressed, we need lots of glucose. And so the epinephrine will provide that for us. It will go to the liver cells, increasing cAMP in the liver cells, and the cAMP is going to activate the enzymes that lead to glycogenolysis and gluconeogenesis. While in the adipose tissue, epinephrine will stimulate the enzymes that lead to the breakdown of triglycerides. Now, GI protein-coupled receptors, GI stands for inhibitory. So these are going to inhibit adenylate cyclase. So activation of the GI inhibits adenylate cyclase enzyme, so that leads to the decrease in the production of cAMP in the cell, and therefore we have inhibited the whatever metabolic processes were occurring in that cell. Now for our second option for these G proteins, we're the opening up of ion channels, and we'll see how that happens. So going back to our scenario, first messenger attaching to the receptor changes shape. That would lead to the activation of what we call here a GQ protein. Q actually stands for nothing. Okay, so they pretty much ran out of letters and they just used that letter Q. So Q just means that we're going to open up an ion channel. Now the activation of that GQ protein leading to the detachment of the protein and then the separation of the alpha subunit will activate this enzyme inside of the cell membrane known as phospholipase C. Phospholipase C will change PIP2. PIP2 stands for phosphatidyl inositol biphosphate. PIP2 is a phospholipid that is normally found within our cell membrane. Activation of phospholipase C will convert PIP2 into IP3 and DAG. IP3 stands for inositol triphosphate, and DAG stands for diacylglycerol. Now, these two are your secondary messengers. These two chemicals have the ability to leave the cell membrane, causing changes within the cell. So let's discuss DAG first. DAG leaves the cell membrane and starts activating different proteins using a kinase known as kinase C. And we'll see what C stands for in a minute. Okay. Again, that will lead to activation of other proteins, leading to your cell response. While IP3 also diffuses and enters into the cell cytoplasm, and it activates ion channels, calcium ion channels that are found on the smooth ER. So remember from a previous chapter, we said that smooth endoplasmic reticulum stores calcium. Sometimes we need that calcium, for example, during muscle contraction. And IP3 would stimulate and open up the gates to release calcium from the smooth ER into the cytosol. So IP3 increases cytosolic calcium levels. Now that calcium could could be your response. Yet on the other hand, it could also be act as a second messenger where it assists DAG in activation of the protein using that enzyme kinase C. So C stands for calcium. Okay. So as we mentioned, that your ion channel can be the effector protein for a G protein. Okay, the G protein can interact either directly with an ion channel, and we really did not see that in this scenario. What we saw is the indirect interaction with an ion channel, where there was a second messenger, which was in this scenario IP3, that opened up the ion channel. So in this scenario, the G protein is called GQ. Okay, the GQ is going to activate a plasma membrane enzyme known as phospholipase C. Phospholipase C breaks down PIP2, that stands for phosphatidyl inositol biphosphate. It breaks down that chemical PIP2 into DAG or diacylglycerol and IP3 or inositol triphosphate. Now, DAG and IP3 are your second messengers that enter the cytoplasm, and they lead to different changes. So DAG is going to activate an enzyme known as protein kinase C, and C stands for calcium because remember it helps in this scenario. While IP3 will attach to a receptor found in the smooth ER that opens up the ligand-gated calcium channels, releasing calcium from the smooth ER into the cytosol, leading to an increased cytosolic calcium concentration. That increased calcium concentration in the cytosol can be your cell response, yet on the other hand, that calcium can also act as a second messenger and assist DAG in the activation of protein kinase C. Now this table is amazing because it has all of your second messengers, where they came from, and what are their effects on the cell. So this again is one of the very important tables for this chapter. So we'll go over them individually. So calcium came from the smooth ER. Remember, we have to open up those gates through IP3. And then the effect on the cell, it helps in the activation of protein kinase C, and it also attaches to calmodulin. We'll talk a little bit more in detail about calmodulin when we get to the muscular system. Now, C-AMP is another second messenger, and that was due to the activation of the GS protein type, activating the adenylyl or adenylate cyclase enzyme, and your effect is the activation of a kinase known as protein kinase A. So protein kinase A stands for C-AMP. Cyclic GMP is also a second messenger, and it was generated due to the activation of the guanylyl cyclase enzyme, and then on its turn activated protein kinase G. So G again stands for GMP. DAG, or diacylglycerol, that was formed, DAG and IP3, both of them were formed due to the breakdown of PIP2 in the cell membrane due to the action of the enzyme phospholipase C. That DAG activates protein kinase C with the help of calcium. IP3, remember we just talked about how it was made. IP3 will attach to the calcium ion channels in the smooth ER, leading to the release of calcium, increasing the cytosolic calcium concentrations. Now we'll talk about other messengers in the body. These group of messengers are collectively known as eicosanoids, and they are formed from a phospholipid found inside of the cell membrane known as arachidonic acid. So we usually have phospholipids within the cell membrane, and we discussed that when we were talking about the structure of a cell membrane. Under certain circumstances, for example, during inflammation or infections, that arachidonic acid will change into these eicosanoids. So these are really stress signals that will be released by the cell when it's damaged or inflamed or infected or burned or whatever kind of damage that's occurring to that cell. The eicosanoids include the family of prostaglandins, thromboxans, and leukotrienes. And you see that they're all in plural. Okay, so there is not one prostaglandin, there are like tons of prostaglandins. Now, these again are considered stress signals that will come out of cells when they are something stressful is going on. And there are two different pathways to form it. So I'm actually going to go to the breakdown of the pathways and then go back to that one slide. So in the case of inflammation, okay, so that first messenger could be a stress signal coming maybe from another cell or coming from, your immune system telling the cells that there's something wrong going on that will activate the receptor, and that would lead to the activation of an enzyme known as phospholipase A2 that will break down your phospholipid and change it into arachidonic acid. Now, arachidonic acid has two pathways depending on what's going on. So if there's an allergy going on, arachidonic acid through a pathway known as the lipo-oxygenase pathway can start making leukotrienes. Leukotrienes mediate allergies and inflammatory reactions, for example, asthma as well. And that is why you can give allergy patients, especially in severe allergy or asthma patients, you can give them these what are known as anti-leukotrienes. For the generic name for the most famous one is Singulair, but the generic name is Montelukast. And again, these are anti-leukotrienes that block the formation of leukotrienes in the attempt to block the allergy and the inflammatory reaction from happening. So that's one pathway. Your other pathway would be the cyclooxygenase pathway, and that leads to the production of prostaglandins that would mediate vascular actions and inflammation, or would lead to the formation of thromboxans, and that would lead to blood clotting and other vascular actions. Okay, obviously it could lead to the production of both, and that's usually what happens in most scenarios. So going back to that one slide, as I mentioned, there are two different pathways. There's a cyclooxygenase pathway leading to the production of prostaglandins and thromboxans, and that is more towards inflammation, while the other pathway is the lipooxygenase pathway, and that would happen more in allergies, leading to the formation of leukotrienes. As you can see, that most these eicosanoids are all in plural. Okay, so we've got tons of prostaglandins, and PG stands for prostaglandins. So there's prostaglandin A, for instance, E, and even the E type has types 1 and 2 and so on. So there's subdivisions of each one of these. Now, how can we stop? We already kind of talked about the anti-leukotrienes, but what about the cyclooxygenase pathway that will lead to the production of prostaglandins and thromboxans? Is there a way to block that pathway? Yes, there is. So we can give what are known as NSAIDs, or non-steroidal anti-inflammatory drugs. So these drugs stop inflammation, but they are not steroids. Okay, so they're non-steroidal anti-inflammatory drugs. One of the most infamous of those are aspirin. Aspirin inhibits the cyclooxygenase pathway and therefore would block the production of prostaglandin and thromboxans. Okay, we are going to use these NSAIDs to reduce pain, fever, and inflammation, so all of the signs and symptoms of inflammation. And again, we call them non-steroidal in order to distinguish them from steroids. So steroids is a very powerful anti-inflammatory drug. We tend to use them in more extreme cases, and we keep the NSAIDs, or the non-steroidal anti-inflammatory drugs, for the less severe cases. Now, how do we stop this whole signal transduction pathway? Okay, the cell at some point needs to stop responding to that ligand. So there are different ways that can happen. Either the ligand disappears, okay, and that could be by the ligand just diffuses away from the cell. It could be that it was metabolized maybe by different enzymes, or the ligand was actually taken up by cells. Well, what if we cannot get rid of the actual ligand? Well, again, remember, the cell has a way to change the number of receptors. Okay, so it can either upregulate or downregulate the receptors. It also has a method to change the shape of the receptors so that now it's no longer a fit for the ligand. So it can deactivate its own receptors. Or we would have to go and change the concentration of the second messenger. Okay, like we did when we were talking about CAMP, how we decreased the level of CAMP using the phosphodiesterase enzyme. So the signal transduction pathway, in order to stop it, could happen at 3 different levels, either at the level of the ligand, the level of the receptor, or the level of the second messenger. And we want to do that in order to prevent chronic overstimulation of the cell. Well, this slide talks about what we can do at the level of the receptor, where we can the cell can chemically change what the receptor looks like, and that would decrease the affinity of the receptor to the ligand, so it is no longer a good fit for that ligand. It can also remove the receptors from the cell membrane through endocytosis. Now know that each cell does not get one chemical messenger at a time. These poor cells are exposed to tons of messages all the time. These messages can really be mixed messages. So it could get stimulatory and inhibitory at the same time, and it's really up to the cell to kind of figure out what is going to happen, what is the net result of all of these stimulations and inhibitions. How am I going to change, and how am I going to control exactly what's going on? Okay, and that depends on the situation. So am I under stress? Am I asleep? Okay, so, different scenarios, different results at the end. So there's a lot of interactions of these signal transductions and a lot of crosstalk that occur in order to figure out what would the end result be. And that this concludes the third and last part of Chapter 5.